Welcome to this lecture on the classification of hormones, a crucial topic in your human physiology course. Imagine you are running a massive, bustling city, and you need to send messages to different factories to tell them what to do. In your body, this communication network is the endocrine system, and the messages are called hormones. Today, we are going to build your understanding of this system from the ground up. We will explore what these chemical messengers are made of, how they travel, and why their classification is the key to understanding how our entire body stays coordinated. Now that we understand the basic premise of hormones as messengers, let us look at how this network operates. Why does the body need different types of messages? Well, some physiological changes need to happen instantly, like a quick text message read by a security guard. Other changes require deep, long-lasting structural modifications, much like confidential blueprints hand-delivered to a boss inside an office. Because of these different needs, not all hormones are built the same way. The body classifies these chemical messengers based on two main factors: what they are structurally made of and how they interact with water. This classification ultimately decides exactly how they will deliver their crucial instructions to the target cells. Building on the idea that hormones are built differently, let us examine their actual chemical structures. Structurally, hormones fall into three main categories. First, we have peptide or protein hormones, which are essentially chains of amino acids linked together. Second, we have steroid hormones. These are entirely different because they are derived from cholesterol, giving them a unique ring-like structure. Finally, we have amine hormones, which are much smaller and simpler. They are modified from single amino acids, most commonly tyrosine or tryptophan. Understanding these three structural categories is essential because, as we will see next, the physical shape and makeup of a hormone directly dictate how it behaves in the watery environment of the human body. These structural differences we just discussed lead us directly to a more critical functional classification: solubility. Why does solubility matter so much? Because our blood is mostly water, but our cell membranes are made of fat. Water-soluble, or hydrophilic hormones, like insulin and most peptides, dissolve easily in the watery blood plasma. However, they bounce right off the fatty cell membrane, meaning they must bind to receptors on the outside of the target cell. Conversely, lipid-soluble or lipophilic hormones, such as testosterone and estrogen, face the opposite challenge. They cannot dissolve in blood, so they require special carrier proteins to travel. But once they reach a cell, they effortlessly slip right through the membrane to bind with receptors deep inside the cell nucleus. So, we have established how hormones travel and bind. But why is this classification so important for the gland itself? The chemical class of a hormone dictates its entire life cycle in your body, starting right from how it is synthesized and stored. For example, because steroid hormones are lipid-soluble, they simply can't be stored in watery vesicles inside the endocrine gland. They would just slip right through the vesicle membranes. Therefore, they must be synthesized strictly on demand. This fundamental structural limitation means that steroid hormones have a slower onset of action compared to peptides, but their physiological effects are much longer lasting. This structural classification extends far beyond the body's internal processes. It is absolutely critical in modern medicine for drug design and administration. If a doctor prescribes a protein hormone, such as insulin for a diabetic patient, it must be administered via injection. Why? Because if it were swallowed, the harsh stomach acids would digest the protein hormone, just like a piece of meat, rendering it completely useless. On the other hand, steroid hormones, such as those found in birth control pills, can be taken orally. Their robust cholesterol-based structure survives the digestive tract and easily absorbs through the intestinal walls into the intestinal bloodstream. Now that we understand the types of hormones and their clinical importance, let us look at the mathematics of how they actually bind to their receptors. The interaction between a hormone and its receptor is not permanent. It is a reversible chemical reaction that reaches an equilibrium. We quantify this using the fractional receptor occupancy equation. In this formula, Y represents the fractional occupancy, which is the proportion of total receptors currently bound to the hormone. It ranges from 0 to 1. The variable H in brackets represents the concentration of the free hormone in the blood. Finally, Kd is the dissociation constant. This is a crucial metric. It represents the exact hormone concentration at which half of the receptors are bound. If a hormone has a very low Kd, it means it has a very high affinity for its receptor, requiring only a tiny amount of hormone to trigger a massive cellular response. This formula we just saw leads us to an important practical application. Let us calculate the receptor occupancy for a newly discovered water-soluble peptide hormone that regulates blood sugar. We are given that the dissociation constant, Kd, is 4 × 10 to the negative 9 molar. After a large meal, the hormone concentration, H, spikes to 1.2 × 10 to the negative 8 molar. We want to find the fractional occupancy, Y. We start by substituting our known values into the numerator and the denominator of our formula. To add the values in the denominator, we align their decimal places, giving us 1.6 × 10 to the negative 8. Because the numerator and denominator share the same order of magnitude, the exponents cancel out beautifully. Dividing 1.2 by 1.6 gives us a final answer of 0.75. Physically, this means 75% of the target cell's receptors are actively engaged in signaling the cell to lower blood sugar. Having seen how hormones bind to initiate a signal, we must now ask, how long does that signal last? This brings us to the concept of hormone half-life and clearance. The chemical classification of a hormone heavily influences its half-life, which is simply the time it takes for its concentration in the bloodstream to decrease by exactly half. Because water-soluble hormones float freely in the blood, they are quickly filtered and broken down, giving them very short half-lives. This allows the body to make rapid, minute-to-minute adjustments. In contrast, lipid-soluble hormones are safely bound to large carrier proteins, protecting them from rapid clearance and resulting in much longer half-lives, which is ideal for sustained, long-term physiological changes. To understand exactly how fast a hormone is cleared, we rely on a specific mathematical model. We model the clearance of the hormone from the bloodstream using the first-order exponential decay formula, which is a standard principle for most drugs and hormones. Let us break down the symbols. C of T represents the final concentration of the hormone remaining in the blood after a certain time has passed. C0 is the initial concentration of the hormone when we start our timer. The mathematical constant E is raised to the power of negative K times T. Here, K is the decay constant, which represents the specific rate at which this particular hormone is cleared, and T is the time elapsed. As time T increases, the negative exponent grows, causing the overall concentration to decay exponentially toward 0. Before we move on, let us see this exponential decay with a concrete example. Suppose we have a water-soluble amine hormone with a known decay constant, K, of 0.0693 per minute. The initial concentration in the blood, C0, is 100 nanograms per milliliter. We want to find the concentration remaining after 20 minutes. We start by substituting our initial concentration, decay constant, and time into the exponential decay equation. Multiplying the decay constant by the time gives us an exponent of negative 1.386. Evaluating e to this negative power gives us 0.25, which is exactly 1/4. Multiplying 100 by 0.25 gives us a final answer of 25 nanograms per milliliter. Physically, this makes perfect sense. The half-life is 10 minutes, so after 20 minutes, 2 half-lives have passed, dropping the concentration from 100 to 50 and then to 25. Now that we have calculated how hormones bind and clear, let us visualize these mechanisms side by side. On the screen, you are looking at a schematic comparing water-soluble and lipid-soluble hormones. On the left side, notice the blue water-soluble hormones floating freely in the red blood vessel. Because they cannot pass through the cell membrane, you can see them binding to the Y-shaped receptor on the outer surface, which then triggers a second messenger cascade inside the cell. Now, look at the right side. The yellow lipid-soluble hormones are attached to large carrier proteins to travel through the blood. However, once they reach the cell, they detach and effortlessly cross the cell membrane. Follow the dashed arrow. They travel all the way into the nucleus to bind directly with the DNA. This diagram perfectly illustrates how a hormone's chemical structure dictates its entire mechanism of action. We have covered a lot of ground today, so let us reinforce the most important ideas from this topic. First, remember that hormones are structurally classified into peptides, steroids, and amines. Second, this structure determines whether they are water-soluble or lipid-soluble, which is the ultimate key to understanding how they travel through the blood and where their receptors are located. Third, we learned that we can mathematically quantify how strongly these hormones bind to their receptors using the fractional occupancy equation. Finally, we saw that a hormone's classification allows us to predict its half-life and how quickly it is cleared from the body. By mastering these concepts, you now have the foundational tools to predict the behavior and mechanism of any hormone in the human body. Excellent work today.